So we like to do the respiratory system after the cardiovascular system because they're so intimately connected to each other. So there's an entire circuit, the pulmonary circuit of the circulatory system dedicated to supplying blood to the respiratory system to pick up oxygen. We're going to talk about that as well as all the other aspects of the respiratory system and hopefully get a better understanding of the importance of the gases that circulate in our blood. So first we're going to go through the anatomy of the system and the functions of the different parts, and then we'll go through how breathing works and how gas exchange works. So the overall functions of the respiratory system are to supply the body with oxygen, to allow for carbon dioxide to be removed from the system. And as we'll talk about, especially with carbon dioxide, the respiratory system has a major role in helping maintain the acid-base balance of the body. Now, there are some additional functions of the system that they're kind of like bonus functions. So they're all important in their own way, but these are the important ones. Like these are the ones that we require for life, and then everything else is just useful. So our sense of smell, as we talked about before, is located within the respiratory system in the nasal cavity. Our ability to speak is mostly due to respiratory function, although there's some other factors involved too. We have a variety of other actions that involve breathing. So coughing, sneezing, hiccuping, and laughing are all weird things we can do with our breath. And then another thing we can do is called bearing down or straining, which is basically where you hold your breath and increase the pressure in your abdomen. So we'll actually talk about that one when we get to the digestive system. So this is the basic anatomy. We start up here in the nasal cavity with the nostrils, and then we go down into this region called the pharynx, and then air travels through the larynx, trachea, and then we've got a bunch of airways into the lungs. So that's how we're going to follow the anatomy, starting with the way that air gets in, and then it'll go all the way in, and then it comes back out the same path. We're not going to follow it back, though. Just go with it from there. So structurally, we divide up the system into two parts. So there's the upper respiratory tract and the lower respiratory tract. And basically, the upper is everything in your head and the beginning of your neck, so the nasal cavity, pharynx, and larynx. And then the lower is everything in your chest. So the trachea is the only one where it's a little bit confusing because it starts up here and then it goes down here. But most of it is in your thoracic cavity. And then your airways are called bronchi, and then your lungs, we'll talk about the rest of that. So we distinguish them for a variety of purposes. We definitely have different things that will affect one and not the other. And so usually we want to keep that distinction. When we look at the functions, though, of the system, the division is very different. So the functional anatomy divides the system into two zones, and they're based on whether or not gas exchange can occur. So the conducting zone is every single structure that is not directly involved in gas exchange. So literally, they're just conducting air between the outside world and the gas exchange. Gas exchange occurs in what's called the respiratory zone, or the respiratory zone. Pronounce it either way. And so this is just the structures involved in air exchange. Now, the interesting thing about the respiratory zone is that it's all microscopic structures, and they're all located in the lungs. So the alveoli are the very smallest things in the lungs, are the end where gas exchange mostly occurs. And then the air gets into there by the very smallest of the airways, which are called bronchioles. Every other structure is just part of the conducting zone. So that's the functional anatomy, and it's much more relevant when we look at gas exchange. as our primary focus. So the conducting zone's functions then, since gas exchange are not its function, is to filter any debris or pathogens from the incoming air. It also is in charge of warming and humidifying the air so that it doesn't damage the tissues in the respiratory zone, which are very delicate. So we want that air to be fairly moist, as well as about body temperature by the time it reaches those alveoli. The parts of the system that are in the conducting zone are, of course, the nose and nasal passages, the pharynx, the larynx, the trachea, and also most of the larger airways within the lungs, which is called the bronchial tree. So the nose does a lot of the functions of what I just described for the conducting zone. It's also our entry as well as exit pathway for air. It's not actually our only one because we are capable of breathing through our mouth as well, but it is the primary one. Its purpose is to allow air movement. So the moistening and filtering and humidifying all start in the nasal cavity. So we have various things in there that filter the air as well as moistening it and warming it. Additional functions of the nose include location of sense of smell. So our olfactory receptors are at the back of our nasal cavity, and then it serves as a resonating chamber for speech. So basically, one of the reasons why our voices sound the way they do is because we have a lot of air-filled structures in our heads, and if they are not filled with air, we sound very different. So when somebody's nose is clogged, like if they're congested, then we describe it as like a nasally sound, and that's because we don't have that air resonation factor. So some diagrams of a bunch of features of the nose that we're not really going to worry about. So basically the nasal cavity runs all the way through here. So there's a lot of air-filled space there. And the smelling part is up here. And once air passes through here, a lot of the particles in it have been filtered out. It's much moister, it's much warmer. It still is going to get more of that as it goes through, but now it's hitting the next part, which is going to be the pharynx. So there is actually a little bit extra to the nasal cavity in the paranasal sinuses. We mentioned these a little bit when we talked about the skeletal system. The paranasal sinuses are air-filled spaces within 4 of the bones of the skull. They are connected to the nasal cavity. So they all have passageways connecting them to the nasal cavity, and they enhance the functions of the nasal cavity. So they produce some of the mucus that helps trap particles. They aid in warming and humidifying the air as it comes in. They help with the resonating function. So they help with phonation, which is how we sound when we talk. And then their other function, and probably their primary function, honestly, is to lighten the skull by removing bone that we don't need. So bone is really heavy, and we have a lot of spaces inside of it so that we don't have quite as much weight to carry around. That's why we also have trabecular bone. That's why we have medullary cavities. All of that is for lightening up our bones. So that's what these guys are for too. But because they're there, they do all these other things as well. So in order to actually accomplish all of these functions, the epithelial lining of the nasal cavity has some features. So there is hair in the anterior portion of the nasal cavity, and along with hair, there are oil glands. The hair functions along with mucus to help trap any larger debris. Once we get past that, because along with this is going to be some stratified squamous epithelium. Once we get past that, we transition to respiratory epithelium, which is pseudostratified columnar ciliated epithelium. You may remember when we talked about tissues that this stuff is really good at making mucus, trapping particulates, and then transporting them with those cilia back up for disposal, so they're not getting farther down the airways. This is a picture of that. So nice tall cells, goblet cells making mucus, cilia, mucus sits on top, you trap stuff in it, and then you transport it. The next part is the pharynx. So the pharynx isn't very long. It's really just back of your throat, but it is a muscular tube. So it is, its walls are composed of skeletal muscle. Its epithelium is continuous with both the nasal and oral cavities, and it participates in several important activities of your throat. So swallowing is your major active function. It does that in cooperation with the oral cavity and the esophagus, and we'll talk about that more when we get to the digestive system. It helps with speech. And mostly the way that it does that is because it is part of the passageway for breathing. So air has to move through the pharynx in order to get down into your lungs. Now, even though the pharynx is not very big, we still divide it into 3 parts. So the nasopharynx is the part directly posterior to or behind the nasal cavity. There are some tonsils in there. Its only function is really the passage of air. And it also contains the openings to the auditory tubes or the nasopharyngeal tubes, which connect the pharynx to your middle ears. So again, that's the guys that pop if you swallow or yawn and you've been like going up or down in elevation. So that's how we equalize the pressure in our middle ears. So that's the first part of the pharynx. The second part of the pharynx is the oropharynx. Oral, like oral cavity, because it's directly posterior to or behind the back of your mouth. So this is a part of the pharynx that's shared between the respiratory and the digestive system, because both food and air have to pass through it. This also contains tonsils, and we will talk about tonsils on Wednesday when we get to immune system. The last part of the pharynx is the laryngopharynx, because it's connected to the larynx, which is the next part in our pathway. It also is shared with the digestive system, so dual purpose for moving air and food, and it contains the entrances to both the respiratory system and the digestive system. So entrance to the trachea as well as entrance to the esophagus. Those are the next steps for both of these systems. So here we have a little bit of a color-coded action. So nasal cavity ends there. This is nasopharynx. Oral cavity is going to end back here. Oropharynx. The larynx is right here. And so all of this back here is laryngopharynx. So the larynx is the connection between the pharynx and the trachea. It is a cartilaginous tube, so there's a bunch of different cartilage structures connected by ligaments forming the walls of it. There are some muscles in there as well. Its job is to regulate the volume of air entering and exiting the lungs by controlling the opening, which is called the glottis. It also contains a feature that prevents inhaling food or liquid when swallowing. So the epiglottis is a flap that closes over the glottis when you swallow. So this is a view of the larynx from the front. Another part of the larynx is the hyoid bone, which you may recall is the only bone in the body not directly connected to any other bone. However, it is connected to the larynx and it's anchored to the tongue. So it's got several important functions. So these are the cartilages. If we look at it from the side in a sagittal section, this is the internal surface. So the epiglottis sits here, and the glottis is this opening here. And then the opening of the esophagus would be back here. And so the laryngopharynx is basically right here. If we look at it head on, so this is what air sees. So, you know, the tongue ends, and then the epiglottis is sitting here when it's open. So the glottis is this opening, and it has several cartilage flaps that can move to open or close this. So these are called the vocal cords or the vocal folds. And this is how we regulate the opening into the trachea, which is back here. The vocal cords are also the most important aspect of vocalization. So our ability to produce sounds is because the vocal cords are able to vibrate as we pass air across them. This is showing you what it looks like when the glottis is open, because the vocal cords are open. This is what it looks like when they are closed. This is also what it looks like when we're holding our breath. So if you take a deep breath in and you hold it, your mouth is closed, but what's keeping air from moving out of your nose is your closed vocal cords. And movement in and out, as well as vibration, is how we make noise. Unfortunately, we don't have time to really talk about it, but if you're curious and you want to see it, there are some videos of somebody speaking and videos of people singing with a scope passed into their pharynx, so you can actually see their vocal cords working while watching them make noises. So it's pretty cool if you're curious. Once we get past the larynx, we're going to enter the trachea. Trachea is also known as the windpipe because it's a hollow tube for the passage of air. That is its job. Now, as the air is coming, we are still filtering particulates. We're still warming and moistening the air. And the other thing about the trachea is that it's not a rigid tube. Like, it's not like you have a PVC pipe in there. So it's actually formed of C-shaped cartilages connected to connective tissue. And you probably have some inkling of that because you can feel your trachea at the front of your neck. But it feels like it's made of rings, and instead they're actually C-shaped and they're open at the back. And connecting across that back part of the C is called the fibroelastic membrane, which is a muscle, a smooth muscle called the trachealis muscle, as well as elastic connective tissue. And those enable the trachea not only to compress a little bit when we swallow, because the esophagus sits directly behind it, but it also allows us to some extent to control the size of the opening of that airway. So here's the trachea. Larynx is up here, as you can see, and then trachea goes all the way down until it branches. Once it branches, it's not the trachea anymore. And then we've got one more view here. So this is a cross-sectional view so that you can see that it really is a C-shaped cartilage with this membrane across it, and the esophagus sits directly behind it. So this is why, because when we swallow, this expands, and so we want it to be able to expand without damaging our trachea. And we don't want our trachea to block our esophagus. So that's why it works like that. All right, so the trachea ends in what's called the bifurcation. It just means it splits in two. And there's a little ridge down there called the carina. The carina is very sensitive, so any particles that make it all the way down there will trigger very violent coughing because it's kind of your last chance to keep things out of the lungs. You'll cough if things get farther in, but like this is the best time to do it. So once we branch, we enter what's called the bronchial or respiratory tree. So we're starting with what are called the primary bronchi. So we have a right primary bronchus and a left primary bronchus. Bronchus is the singular, bronchi are the plural. So one of them is the entrance to the right lung, obviously the right one, and the other one is the entrance to the left lung. And then they branch from there with the airways becoming smaller and smaller. When we get to the smallest branches, that's when we transition into the respiratory zone from the conducting zone. And we see changes in the structure of these airways as we get smaller and smaller because they have slightly different roles. So the primary bronchi have complete cartilage rings, but then when things get super small, rings aren't going to cut it. So we just have these little plates to help hold the airways open. And then the last ones are so small, there's no cartilage. They just have elastic fibers. So they're able to stretch, but there's no rigid structure keeping them open. We transition our epithelium from a pseudostratified columnar to a simple columnar down to a cuboidal because we're getting smaller and smaller and smaller, and we no longer need the same types of secretion and protection functions. And we see an increase in the amount of smooth muscle in the walls of these airways. And that's because the smaller airways have a greater ability to change their diameter to change how much air is moving in. So this is what that looks like. So trachea branching into the primary bronchi, and then they just keep branching from there. And you can see they get very, very small, and different segments of the lungs are supplied by different branches. Now, the crazy thing is that it really does look like that. Like that is a real, like that was a computerized image, but it was based on like actual scans. This is actually a giant blood clot that filled the airway, clotted, and then somebody coughed it out at a hospital, which was kind of crazy. So I like to include that, even though it's slightly horrifying, because it does look like that. So that takes us to the respiratory zone, which again are the structures that directly participate in gas exchange. These are the things that are small enough and thin enough for diffusion of gases to occur. So that includes the respiratory bronchioles, which is what we call the very smallest bronchioles. That leads us to alveolar ducts, which connect us to the alveoli. So an alveolus is the end sac of airways in gas exchange. It's a dead end, or a cul-de-sac, if you will. And they're actually all connected together. They tend to be clustered like little grapes. So the cluster is called an alveolar sac, and there's little alveolar pores connecting them all to each other as well. So even though they're dead ends, air doesn't necessarily get trapped because it can move to another one, which is literally what that's for. So this is how we represent that. So the last bronchioles, they, instead of cartilage now, we just have smooth muscle. So their diameter can be modified. So terminal bronchiole into alveolar duct, into an alveolar sac of a bunch of different alveoli. And these are showing cuts. You can see the pores connecting everybody together. And then this is showing you the blood supply. We have deoxygenated blood from the heart coming in, picking up oxygen, and then oxygenated blood going back to the heart. So this is coming from the right ventricle, and then this is going back to the left atrium. Talk about that a little more later. We have a couple different types of cells in the alveoli. They all have different functions. So the majority of the alveolar walls are made of type 1 alveolar cells. They are simple squamous epithelium, which remember is as flat as it goes. So they're these flat little cells with the nucleus, and they're flat because we want them very thin for gas exchange. Because remember, the advantage of a squamous epithelium, of a simple, is diffusion. Easy to have that occur across a very thin cell. There are type 2 alveolar cells, which are not flat. They produce a product called surfactant, which is important for keeping the alveoli open. And we'll talk about that in a little bit. The alveoli are surrounded by elastic fibers, which allow them to expand when they fill with air and then gently close again when they get some air out. And then the last big thing we see here are alveolar macrophages. So macrophages are part of the immune system. and they phagocytize anything that actually makes it all the way down into the lungs, debris and pathogens. And then as I showed you, these guys are surrounded by capillary beds of the pulmonary circuit. So deoxygenated blood in the arteries, picking up oxygen, and then oxygenated blood in the veins back to the heart to go to systemic circulation. So in order for gas exchange to occur, we need to move the gases not only across those simple squamous type 1 alveolar cells, but into the blood. And so the total thickness of cells across which the gas exchange is occurring is called the respiratory membrane. So it's composed of those simple squamous type 1 alveolar cells, a basement membrane, because all epithelial tissues have a basement membrane, and then the simple squamous endothelium of the capillaries. So we're going across two simple squamous epithelial layers with a basement membrane in between. It's half a micron thick. That's micrometer, very thin. And so the respiratory membrane is also called the blood-air barrier because it's the only thing between the air in the alveoli and the blood. So very different from the blood-brain barrier, which is quite thick and trying to keep things out. This is all about making things easy so gas exchange can occur readily. So here's another diagram of the respiratory zone with capillaries. We can see the capillaries here. So we've got endothelium and the type 1 alveolar cells. So this is the respiratory membrane, just there, just that thin little thing. All right, does anybody have any questions? Because that is the anatomy of the pathway that air takes. All right, so now we're going to look at the anatomy of the thoracic cavity that the lungs are actually sitting in so that we can talk about how air actually moves in and out. So lungs, major organ of the system, housing the majority of the conducting zone and the entirety of the respiratory zone. Function is, of course, to exchange oxygen and carbon dioxide. Now, the way that it does that effectively is that it actually has a very, very large surface area, in addition to having just a very thin or small distance for diffusion to occur. So the large surface area is because even though lungs fit inside your thoracic cavity, if you laid out all those little alveoli, you'd have a very large area. And large surface areas are crucial when we're relying on diffusion for the exchange of materials. So we have a right and a left lung. Hold on, I can't remember if I have, nope. We have a right and a left lung. They are divided up into lobes. So the right lung has 3 lobes, superior, middle, and inferior. The left lung has only 2 lobes, superior and inferior. The left lung also has a little notch in it because the heart takes up that space. So that's your basic anatomy. There is a dual blood supply to the lungs as well. So we've talked about the pulmonary circuit supplying the alveoli. We know that the right side of the heart is pumping the blood into the lungs by the pulmonary arteries that were exchanging gases at the pulmonary capillaries and that were returning blood to the left side of the heart by the pulmonary veins. But there is systemic circulation into the lungs as well. And those are the bronchial arteries from the systemic circulation. And that's because the larger airways are too thick to get gas exchange just from what's inside. and they also need some amount of nutrition as well. So basically, we need to provide oxygenated blood to the larger airways. Innervation, so the way that the nervous system is able to control the airways and stuff. This is mostly about airway diameter, so smooth muscle control, is by the pulmonary plexus. So a bunch of nerves that supply all the airways so that we have autonomic control of airway diameter. When we look at the thoracic cavity as a whole, we see that each lung is set up very similarly to the heart. So there is a serous membrane around each lung, enclosed in a pleural cavity, which is within the thoracic cavity. So we look at the whole thing, and the heart's in here, in the pericardium, and each lung is separated into a pleural cavity. There is a visceral pleura that lines the outside of the lungs, just like we have a visceral pericardium on the outside of the heart. And then there's a pleural cavity with pleural fluid, and then lining the inside of the chest wall is the pleural, or the parietal pleura. So the parietal pleura, sorry, that should be pleura, not plural, lines the thoracic wall, so the inner part of your ribs and muscles, the mediastinum, which is the central part where the heart sits, and then the diaphragm as well, because the lungs are sitting against that skeletal muscle. So just like the pericardium, this is very much like a fist going into a plastic bag with a little bit of water, and so that overlap occurs on the medial side at what's called the hilum of each lung, which is where everything goes in and out of the lungs. So if we look at it like this, we're kind of looking, so we've got the heart's missing here. You can see the left lung here. The hilum is right here where the primary bronchus comes in. All the blood vessels go in and out of there as well, as do the nerves. And so the parietal pleura surrounds the lung, attached to the wall and the diaphragm and the tissues of the mediastinum. And then the visceral pleura is attached directly to the outside of the lung. So here's a close-up over here. Visceral pleura is attached to the lung, little tiny pleural cavity, parietal pleura attached to the chest wall. All right. So each lung has its own, and they're set up in the same way. And then this one gives you a nice view of the diaphragm, which is a sheet of skeletal muscle that actually attaches to your vertebrae in the back. And then your ribs up here. And that takes us to how we actually breathe, because we need to understand the pleura and the pleural cavities and the diaphragm to understand how breathing actually works. So we're going to go through that one step at a time, and then we'll talk about gas exchange. So pulmonary ventilation is the technical term for breathing, and it is a two-phase cycle. So inspiration is what we call inhaling. I don't know why we have two different words, but we do. And then expiration is what we call exhaling. When we're talking about the movement of air, it's always about pressures and volumes of gases. And we look at the movement of air into and out of the lungs relative to atmospheric pressure, which is the pressure of the gases surrounding us. That is measured as 760 millimeters of mercury is equal to 1 atmosphere. This is basically how much air sits on you when you're at sea level. So we'll talk about some negative pressures when we look at how air moves in and out of the lungs. And it doesn't mean that the pressures are actually negative. It just means they're lower than atmospheric. Hopefully that'll make sense as I go through it. So with gases, the pressure of a gas and the volume of a gas in an enclosed space are inversely related. So basically gas has, you know, molecules just suspended, and they can be really, really spread apart, or they can be closer together. So if we take a certain number of particles of gas and we squish them together, we can decrease the volume, but we're going to increase the pressure. And that's because the pressure is basically just determined by how much these particles are bouncing off the walls. So just like pressure of blood is about how much the blood is pushing against the walls of the blood vessels, pressure of gases, like air, is about how much the particles of the gases are hitting whatever is enclosing them. So if we decrease a volume, we're squishing all these particles closer together, and now there's a relatively larger number of particles for the space that they have. So now there's more of them bouncing against the walls, so we have a higher pressure. If we do the opposite, if we increase the amount of space that they have, then we'll get a decrease in pressure because now there's fewer particles for the amount of space, which means fewer particles bouncing off the walls. We can do it the other way around too. We can increase the pressure to decrease the volume, and we can decrease the pressure to increase the volume. So we can change either one, and we'll see this change in the other. And inversely just means that they change in different directions. So decreasing the volume, increases the pressure, increasing the volume, decreases the pressure. So we actually represent that with something called Boyle's law. This means the exact same thing that I just said. So this says that a pressure times a volume is equal to a change in that pressure times a change in that volume. So this is how we can represent that visually. This is showing you increasing the volume and decreasing the pressure. And then this one is showing you decreasing the volume to increase the pressure. So you can see it's the same number of particles, but here they're more spread out and therefore bouncing off the walls less. Here they're closer together, so they're bouncing off the walls more. Atmospheric pressure is literally the column of gas above you in the atmosphere. So our elevation actually affects our atmospheric pressure. Now, we're not going to go into too much detail about that, but just to kind of help you understand the whole concept, I like to show this. So when you're high on a mountain, you have less gas pressing on you, so your atmospheric pressure is lower. When you go back down to sea level, the column of air is much larger, and so your atmospheric pressure is higher. There's more gas pressing on you. If you went somewhere that's below sea level, whether you're under the sea or whether you're just somewhere like Death Valley that's actually below sea level and still land, the pressure is even higher because now there's even more of a gas column. But when we consider atmospheric pressure, we usually consider sea level because sea level is our standard. It's like room temperature has a standardized value of like 72 degrees, might be 74, doesn't matter. So when we say 1 atmosphere, we always mean sea level, which is 760 millimeters of mercury. So the respiratory pressures that we are then comparing to atmospheric, there are 3 of them. So intrapulmonary pressure is the pressure inside the alveoli. So how much gas is inside of the alveoli? Because unless you actually close your airways, we have a continuous uninterrupted path between the outside and those alveoli. We're always going to equalize this intrapulmonary pressure with the atmospheric. And we're going to do that by having the gases flow down their pressure gradients just the way fluids do, like we talked about in the last chapter. So we're always going to flow from high pressure down to low pressure. The second pressure is intrapleural pressure. So this is the pressure of the pleural cavity, that small little space between the outside of the lungs and the inside of your thoracic wall. This is always negative, as in it's less than atmospheric pressure, and it stays 4 millimeters of mercury below the intrapulmonary pressure. So it has to always be negative, and we'll get to why that is in a second. When we look at those two values together, we get a combined pressure of transpulmonary pressure. So transpulmonary means across the lungs. So this is the difference between the intrapulmonary and interpleural pressure. And this should always be that negative 4 millimeters of mercury. So it should always be a negative value. And it's necessary because it keeps the lungs open. It keeps the lungs from collapsing. Essentially, this negative pressure keeps the lungs stuck to the inside of the chest wall. And if it's not there, if it's not negative, then those lungs will just collapse in and pull away from the chest wall. So if the transpulmonary pressure ever is 0, so they're equal to each other, then we will have lungs collapse. The most common time for this to happen is if you get an injury through your thoracic cage, and now there's air from the atmosphere getting into your pleural cavity. That equalizes the pressure, and then your lungs collapse. One second. Take a look at this. All right, so this is the visual of what the last two slides were. So the intraalveolar or pulmonary pressure is whatever's inside the airways and the alveoli. The intrapleural pressure is what's inside the pleural cavity, and then the transpulmonary pressure is what's in between the pulmonary and intrapleural. So you don't need to know the numbers, just need to understand the concept. This is also a really nice demonstration of the parietal and visceral pleural membrane. All right, so how does this result in breathing? So when we want to inspire, when we want to bring air into our lungs, we're going to use our diaphragm. So when our diaphragm contracts, it actually pulls down into the abdomen. And by pulling down into the abdomen, we actually increase the volume inside our thoracic cavity. Now we can do this with additional muscles too. So we have external intercostal muscles between our ribs that can lift and expand the whole rib cage. And we have even more muscles if we need to take a really deep breath. But the point of all of them is to increase the thoracic volume. Now, because of that negative pleural pressure, when the thoracic wall increases, the lungs increase in volume too. They follow. They're stuck to there. And so that means that we're increasing the volume of the lungs. So Boyle's law says that if we increase volume, we decrease pressure. And we know that air is going to move down its pressure gradient. So if the pressure inside the lungs is lower than the pressure of the atmosphere, air is going to move into the lungs. So here this is, we increase the volume, that decreases the pressure. So now we move down the pressure gradient into the lungs. Okay, inspiration with active diaphragm contraction at the minimum. Expiration is the opposite process. So usually it's passive. We don't actually have to use any muscle, although we can. We do have muscles that can do this. But usually all we have to do is relax the muscles that were active. Because there's a lot of elastic tissue in the lungs and the rib cage, we have an elastic recoil that decreases the volume of the thoracic cavity. This is going to decrease the volume of the lungs. And as we know, if we decrease the volume, we're going to increase the pressure. once the pressure inside of the lungs is greater than atmospheric, we're going to have air moving out of the lungs and back into the atmosphere. Now, if we need to take a really deep expiration, a deep breath out, we can engage our abdominal muscles to kind of push stuff up into our thorax, as well as our internal intercostals to pull our ribs together. These will enhance the decreasing volume, which therefore enhances the increasing pressure, which moves more air out of the lungs. Okay, so here's expiration. So when we relax that diaphragm, it bounces up and pretty significantly decreases the volume. And so then we have that increase in pressure and out it goes. Here's a different image to get a different view of how the diaphragm moves down when you contract it and then bounces back up. Now, obviously, it's not always that easy. So that's the basics. And so your take home is therefore that skeletal muscles are actually doing the work of breathing. Your lungs don't have muscles in them that allow breathing to occur. So they're just passively expanding and contracting based on the skeletal muscles changing volumes. So the pleural fluid in the cavity helps keep the lungs attached to the thoracic walls, in addition to that negative pressure. And that's because of something called surface tension. So this is the same concept that will like stick something to something else. If you've ever had two pieces of plastic or just a plastic bag with water in it, and you're trying to separate the two layers from each other, it's hard to do because the water is sticking them together. This is the same thing that sticks the lungs to the insides of the thoracic walls. Exhalation is even more passive. It's mostly just relying on elastic recoil. So we have elasticity to our thoracic wall and our lungs, and so when we stretch, they automatically bounce back to their original shape. Now, there is a force that we have to work against in order to achieve breathing. So just like we work against friction when it comes to blood flow, we also have to work against friction when it comes to airflow. In this case, it's the friction of the molecules of the air against the walls of the airways. And so that's what we're working against with those skeletal muscles. Just like blood vessels, the smaller airway diameter is going to have more resistance. More resistance means more of a change in pressure achieved by more of a change in volume in order to get the same amount of airflow. Now there's another force that usually is constant that we also have that we have to work against. And that's the surface tension within the alveoli. So we have to keep the inner surface of the alveoli moist to keep the tissues alive, right? So remember, we went to all the trouble of warming and moistening the air to get it in. But unfortunately, the surface tension that keeps the outside of the lungs attached to the inside of the thoracic wall also wants to pull the walls of the alveoli together. So that would collapse them, and then we wouldn't have any air in there, and then we wouldn't be able to actually do gas exchange, which would be really bad. So in order to keep the alveoli from collapsing, we have to reduce the surface tension. And that's why we have pulmonary surfactant. So surfactants are substances that decrease surface tension. They kind of work like soaps or detergents. And because we have surfactant in our lungs, our alveoli stay open. So this is a little demo showing you, because of course, again, the smaller the diameter, the more likely we are to collapse. So with this one over here, there's no surfactant, we just have water lining these guys. Because this guy is smaller, it has higher surface tension and it would collapse and it would be really hard to open again. But if we add surfactant, then we have less surface tension on this side. So we're going to stay inflated. And don't worry about the rest of what it says. All right, so just like we worry about compliance when it comes to the heart and blood vessels, we worry about compliance in the lungs as well. So lung compliance refers to how stretchy the lungs are, right? So how much expansion or change in volume we can get for a given change in pressure. The more we get, the more compliant it is, and this means less work. So compliance means less work for our breathing muscles. Two different factors are going to determine how compliant our lungs are. So the elasticity or distensibility of our lung tissue, so how well it expands. Normally it's quite high. And then the alveolar surface tension, so the tendency of our small airways and alveoli to collapse because of the water in them, which normally is really low because of surfactant. So anything that alters one of these factors is going to decrease lung compliance and make breathing a lot more work. This is showing you compliance as determined by the volume of air in the lungs and the pressure. So this is normal. With fibrosis, we have a decrease in compliance because the airways and stuff have less elastic tissue and more collagen fibers, and so then they're not as able to expand. And so that means that we have less volume in our lungs, even though we're using much higher pressures. So you're working harder and you're getting less out of it than a normal lung. Emphysema actually shows an increase in compliance, and that's because we lose a lot of the alveoli in the disease of emphysema. On the bright side, your lungs are really stretchy. On the downside, you're not getting as much gas exchange because you don't have as many alveoli. So this is still a bad situation, but working to breathe is not the problem, which is why people with emphysema usually need to be on supplemental oxygen. All right, so when we look at how much air we can move in and out of the lungs, we get something called respiratory volumes, because you all know this, we don't always breathe the same amount of air, right? Sometimes we breathe more and sometimes we breathe less, depending on how much we feel like we need. So respiratory volumes are how much air can be moved in and out of the lungs. Tidal volume is how much you're moving in and out in one breath. Inspiratory reserve volume is how much more you could breathe in if you needed to. So how much more you could breathe in to take a maximum breath in. Expiratory reserve volume is how much more you could breathe out if you needed to. And residual volume is what's always in your lungs. So just like we never empty the heart of blood, we never empty the lungs of air. And so the amount that we cannot get rid of, which is good, we don't want to, we really don't, but the amount that we always have, which keeps our alveoli and small airways inflated, is called the residual volume. It's what's left over when you have breathed out as much as possible. It's the residue. So it looks like this. Take a look at that for a second. Give me a moment. Stay out of it. Very, very naughty. Everybody can hear it. It's just not helping. All right, so we'll just work our way up. Residual volume So this is, you don't need to know these numbers, but just to give you something. So it's about 1,200 milliliters of air that we always keep in our lungs. This is average. It does vary by size of person, okay? But this is average. And up here we have our tidal volume. So this is about how much air we usually move. We usually move about 500 milliliters, so half a liter. This is just normal quiet breathing. And it's tidal because, you know, it's like the tide. If you've ever been to an ocean and the waves come in, the waves go out, it's that very soothing rhythm. That's kind of what breathing is. If you're trying to take a maximum breath in, your inspiratory reserve volume is how much more air you could bring into your lungs if you needed to. So you can see it's how much volume you could add above your tidal volume, which is quite a lot, right? Several liters. And then your expiratory reserve volume is how much more you could breathe out if you needed to. So you can get about a liter more out if you needed to, okay? So definitely a big range of air volumes that we can move into and out of our lungs. It doesn't seem like we'd be able to hold that much, but that's where all that surface area comes in. As far as control, this is where things differ quite a lot from the heart. So there is no pacemaker of the lungs. There's no autorhythmic activity. I mean, the lungs aren't even doing the job, right? It's the diaphragm that's the primary muscle of breathing. And so that means that the lungs are not in charge of the rhythm of breathing. They're not in charge of the rate, and they're not in charge of the volume. Instead, all of that comes from the brain, including the pacemaker. Respiratory rate is, of course, the number of breaths per minute. And that is controlled mostly by the brainstem. So the brainstem controls the breathing rate. It'll also control breathing depth. And it uses a variety of inputs to dictate how fast you should breathe and how deeply you should breathe. So we get sensory input by chemoreceptors. We'll talk about that in a second. There are some respiratory reflexes, and then you can override your brainstem with higher brain centers. So we're going to work through each of those things and see how that works. So this is just showing you your brainstem controlling all of the breathing muscles, because that is, of course, what it would control. The major factors that influence the rate and depth or volume of your breathing are mostly chemical factors. So because breathing is all about moving oxygen in and carbon dioxide out, those are the things that are going to determine if you're breathing appropriately. So we have chemoreceptors in the brainstem and in the aortic arch and carotid arteries, which is the same place that we keep the baroreceptors that monitor blood pressure, interestingly. And that's because those are major places that are always getting a lot of blood. It turns out that the most important thing in determining rate and depth of breathing is carbon dioxide level. We always think that it's about oxygen, that like when you're breathing more, it's because you need more oxygen in, and you know, that's what's driving your breathing, but it's actually about carbon dioxide. And the way that we have a very sensitive monitor of that is by monitoring pH. So carbon dioxide is linked to the pH of our blood, and that's because carbon dioxide is able to combine with water to make carbonic acid. So this happens fairly readily, and because of that, when we have an increase in carbon dioxide in the blood, it ends up meaning an increase in acid levels and vice versa. So we have chemoreceptors in the brain detecting pH changes. And if we have an increase in acid, so that's a decrease in pH, we end up breathing more. So hyperventilation means an increase in breathing, And the point here is to get rid of more carbon dioxide, because whenever we breathe out, we have more carbon dioxide leaving the body. As we get rid of carbon dioxide, we reverse this process and we decrease the amount of acid, so we raise the pH. The opposite occurs as well. So if the pH increases, so that's alkalosis, we stimulate hypoventilation, which is a slowing down of breathing. That lets carbon dioxide build up, which builds up the amount of acid in the blood and will get us back to normal pH. Carbon dioxide has direct impact on respiration as well. So we have chemoreceptors that detect carbon dioxide levels these are the peripheral ones in the blood vessels. So increases in carbon dioxide, which is called hypercapnia, that's just the word for it, is going to increase our respiratory rate, and decreasing levels are going to decrease it. You might think that we'd want to just get rid of as much of it as possible, but we actually wanted to maintain a fairly constant level. Okay, so carbon dioxide controls our breathing. These are feedback loops or reflexes. So pH and carbon dioxide here, they're actually backwards. Oh well, it's fine. Affecting the chemoreceptors, and those are informing the brainstem centers, and that is affecting your breathing muscles, which is how we're controlling our respiratory rate. Now, oxygen is detected as well, but it doesn't have as strong of an influence on our breathing as we think it does. So oxygenation status doesn't affect our breathing unless the oxygen levels in our blood drop below 60 millimeters of mercury. 60 millimeters of mercury is what we call hypoxia, and that is bad. Now, we're normally at about 100 millimeters of mercury, so that's a pretty significant drop. And in a healthy person, it's only going to occur if you go to a high enough altitude that the oxygen levels are very low, and then there's just not as much oxygen in the air, and you'll get below 60 and you'll have what's called, and you'll start breathing very heavily unless you acclimate. So acclimating is slowly working your way up to let your body get used to lower and lower oxygen levels. People that live at really high altitudes might end up being dependent on oxygen levels for breathing. It's called hypoxic drive. So basically, if you never have very high oxygen levels and this is always stimulating your breathing, it can take over. But for the rest of us, it's all about carbon dioxide. And I emphasize that because it's counterintuitive and not how we're used to thinking about breathing. Higher brain center overrides. So the hypothalamus, of course, is capable of modifying the rate and the depth of breathing, triggered by emotions and pain. And then we can see alterations in breathing based on changes in body temperature as well. So we're more likely to breathe more slowly when we're colder and more fast when we're hot. We can also have cortical control. So your cerebral cortex, so voluntary control of the rate and depth of breathing, right? I'm sure you all know that already, and if not, you've been experiencing it throughout this lecture, because there's nothing like talking about breathing to make you think about it and take over control again. Now, the nice thing about this is that whenever you stop paying attention to your breathing again, your brainstem takes over as soon as you become hypercapnic. So as soon as your carbon dioxide levels increase again, and you're not actually bothering to breathe voluntarily, you automatically start breathing without thinking about it again, and voila, off you go. So it's a very nice setup. The other things that can override normal breathing are the reflexes. So respiratory reflexes are all about keeping your airways and your alveoli protected. So the irritant reflexes are the ones that help us get rid of debris, as well as excessive mucus or even fumes, like exhaust fumes or something like that. And that's because the bronchioles are very sensitive to this stuff. So also our nasal cavity is very sensitive. So what we see is a couple of different things. So anything getting down into your lungs will trigger reflexive constriction of the bronchioles to decrease airflow. And then when this goes horribly, horribly wrong, that's how we get asthma. Particulates are going to trigger coughing to try to get out anything. And then if it occurs in your nasal cavity, then you're going to get sneezing. So sneezing is about getting things out of your nasal cavity that are irritating. Coughing is about getting things out of your lungs. We also have something called the inflation reflex, which prevents us from overstretching our lungs. So one of the things that limits our inspiratory reserve volume is this stretch reflex. Basically, we have stretch receptors in the pleura as well as the conducting zones of the lungs. And once they get triggered as we try to breathe in maximally, they will shut off our control of the diaphragm, prevent us from breathing in anymore so that we're not damaging our lungs by overstretching them. All right, does anybody have any questions? Because then we just have gas exchange. All right, so a couple more physics related things. There are a couple more gas laws we need to be aware of in order to understand how gas exchange takes place. So this is just more stuff about pressure. Basically, when we have gas interacting with either a solid or a liquid surface, and liquid surfaces are what we're going to worry about, there's pressure, right? So we know that bouncing against the walls is pressure, but bouncing against like the surface of water also is gas exerting its pressure. And the thing is that when you have a mixture of gases, like we do in the atmosphere, each gas exerts its own pressure independent of the other gases. So for example, you know, relevant, super relevant too, our atmosphere is mostly composed of nitrogen, so 78.6%. You don't need to know these numbers. So mostly nitrogen, almost 21% oxygen, very low carbon dioxide levels, and then water vapor does vary. Just not usually super high, but it is variable, and we're quite sensitive to that variation, but not super relevant to our purposes. So Dalton's law is the one that says that the individual pressure contributions of each gas add up to the total pressure of the whole gas, which means that all of the pressures from each individual component of our atmosphere, which is almost entirely nitrogen and oxygen, add up to the total atmospheric pressure. So at sea level, that's that 760 millimeters. The individual contributions are called partial pressures because they're part of the total. When the total atmospheric pressure changes, the partial pressure changes too, which means that even though the composition of gas stays the same, when we go up in elevation, the total pressure is lower because there's less gas column over us, and that's why there's less oxygen available, because even though it's still basically 21%, now it maybe is 21% of 730 instead of 760, and that can be enough to make it hard for us to get enough oxygen. This is a little visual of that. So particles of oxygen, particles of nitrogen, their atmospheric pressures. When you combine the particles together, you get a combined pressure that is equal to adding these guys together. Okay, that's partial pressures. The second thing we need to understand is solubility. So we know that things can dissolve into solution, right? So solvent is the liquid that is the largest proportion, which for our purposes is water, and solutes are things that dissolve. So gases can be solutes as well. And anytime a gas is in contact with a liquid surface, some amount of that gas is going to dissolve. This is in part going to be dictated by its partial pressure. So a higher partial pressure means a higher proportion of the gas is going to dissolve. And gases tend to reach equilibrium between the gas and liquid phase. So this, for example, is how carbonation works. So with carbonation, we're shoving a bunch of carbon dioxide into a liquid. We're making it dissolve. And then because there's not very much carbon dioxide in the atmosphere, we're capping that liquid to trap it all in there. So then when you release that cap, you get a bunch of carbon dioxide escaping out, moving down its pressure gradient. And if you leave it out uncapped, eventually all the carbon dioxide is going to exit the liquid because it's going to continue to move down its concentration gradient into the atmosphere where there is basically no carbon dioxide. And then you end up with flat soda. So the same thing has to happen in the lungs, but we don't have very much time, right? We have the time of a single inhalation. This is a demonstration of Henry's law. So a bar is the same as an atmosphere. It's just in nautical terms because you usually only find this stuff if you look up things related to scuba diving. Basically, 1 atmosphere gets you that much in the liquid. You double the pressure, you double the amount that dissolves. All right, so there is one other factor as far as solubility, and that's literally how soluble a gas is. So pressure is not the only factor. It's also about how readily a gas wants to dissolve in water. Temperature is also a factor, but because we typically have a constant temperature internally, we're not going to worry about it for ourselves. But generally, more gas is able to dissolve at a lower temperature and less gas at a higher temperature. And that's why we like to have carbonated beverages ice cold, and nobody ever serves warm carbonated beverages. Like, it doesn't happen because you can't keep the gas in like a tea. You know, we don't do warm Coca-Cola for a reason. But again, that one doesn't really matter because it's pretty constant in our bodies. So instead, it's about solubility. And the thing is that carbon dioxide is much more soluble than oxygen or nitrogen. It very readily dissolves into solution. Oxygen is only a 20th the solubility of carbon dioxide, and nitrogen is half as soluble as oxygen. And this is why at a normal atmospheric pressure, even though there's almost 80% of the atmosphere is nitrogen, there's basically no nitrogen in our blood because it's just not very soluble. All right, so although the solubility has a major factor, ultimately what moves gases is their pressure gradients. So we're still going to have gases moving down their pressure gradients, but now it's down their partial pressure gradients. So this is still passive diffusion. Now, in the across the respiratory membrane, we have a very steep oxygen gradient, and that allows oxygen to move quite readily from the air in the alveoli. It does have to dissolve in the water. Water film that sits on the epithelial cells first crosses the respiratory membrane into the blood. We can reach equilibrium, pressure equilibrium, in 1/4 of a second. That's not too bad. Carbon dioxide, of course, is going to go from the blood into the air. It has a much smaller gradient, but the greater solubility means that we still get a decent amount of movement. So gas exchange actually occurs at 2 places. So what I just described was external respiration, which is the movement of oxygen into the blood and carbon dioxide out of the blood in the lungs. But internal respiration is where oxygen moves from the blood into body tissues, and carbon dioxide moves out of body tissues into the blood. So we have 2 places of exchange, and every single time we're exchanging, we're kind of resetting for the next spot, right? Because this is going through the 2 circulatory circuits, right? This is the pulmonary circuit, and this is the systemic circuit. So in external respiration, we're going to have carbon dioxide moving from the blood into the alveolar gas. Oxygen dissolves into the blood, and then it's going to bind to hemoglobin. Partial pressure gradients are the most important factor, and then gas solubility is the second. But there are other things that influence this as well. So the thickness and the surface area of the respiratory membrane. In a normal person, it's plenty for gas exchange to occur. And then ventilation perfusion coupling refers to matching the gas to the blood. And we're not really going to worry about that, but it is a factor as far as making sure that all the alveoli have enough blood and all the blood is surrounding inflated alveoli. So these things are really not super important in a healthy person. Like, we don't see a lot of issues because both of them are sufficient. So when we look at this, we're really just looking at these partial pressures, and this is showing you the movement of this stuff. And I'm going to let you refer back to this after I talk about how oxygen and carbon dioxide are carried in the blood, because honestly, it's more useful for that. Internal respiration, we're reversing this, of course, and we're gaining carbon dioxide in the blood, and then we're losing oxygen because the tissues need it. This is influenced, of course, again, by the partial pressure gradients, but also by tissue perfusion and metabolism. So tissue perfusion refers to how much blood they're getting. Is it enough blood for all exchange to occur adequately? And tissue metabolism refers to how much oxygen they're using and how much carbon dioxide they're producing. Because this literally controls how much of a gradient we have. So we look at the one for internal respiration. So the higher the metabolic rate of the tissue cells, the lower their oxygen levels, which drives oxygen in, and the higher their carbon dioxide, which drives carbon dioxide out. If we don't have very metabolically active tissues, then we're not going to have as much gas exchange because they literally don't have as much of a gradient to work with. So in between internal and external respiration, we have transportation of gases. And this is where we really overlap between the blood, which is technically part of the circulatory system, and movement of gases, which is all about the respiratory system. So oxygen is all about being bound to hemoglobin. Almost all oxygen is carried attached to the ions of hemoglobin, and a very small proportion is dissolved in the blood and just is part of the solution. Very low amount. When oxygen binds to hemoglobin, we call it oxyhemoglobin. And when a hemoglobin has all four ions bound, we call it a saturated hemoglobin. And then when there's no oxygens, we call the hemoglobin deoxyhemoglobin because it's got no oxygen on it. Oops, sorry. Interestingly, when oxygen binds to hemoglobin, it changes the shape of the whole molecule so that it's better able to bind additional oxygen molecules. So that means that, like, when we have a hemoglobin with 3 oxygens on it, that 4th one is going to go on there really nice. But as they lose oxygens at the level of the tissues, the shape shifts back, and we actually have a decrease in affinity for oxygen molecules. And both of these things are really nice because when we're in the lungs and we have lots of oxygen coming in, wanting to bind, the more oxygen we get binding, the more oxygen we're able to bind. And then we get to the tissues and we start losing oxygens again. And the more of them we lose, the more come off. So it really facilitates both internal and external respiration. If you don't remember what hemoglobin looks like, the hemes are where the oxygens go. So this trend of changing the shape of the hemoglobin to change how well it binds oxygen gives us something called the oxygen dissociation. And it's basically how likely oxygen is to bind to hemoglobin based on how much oxygen is already present in the blood. A lot of different factors influence the curve. And again, this is just a nice way to look at how conditions in the lungs facilitate binding oxygen to hemoglobin, and conditions in the tissues facilitate transferring that oxygen to the cells. So it looks like this. Basically, this is fully saturated hemoglobin. So all of the hemoglobin and all the red blood cells have all of their oxygen, have all of their ions bound oxygen. Over here we have the partial pressure. So this is the lungs. The partial pressure is 100 millimeters of mercury, and we're readily able to achieve full saturation. We can even achieve basically full saturation at 80 millimeters of mercury. And then this blood, this saturated blood, goes to the tissues. And then the tissues have a very low partial pressure because they've been using all their oxygen. So they only have 40 millimeters of mercury of oxygen. So this is the gradient down which oxygen flows from 100 to 40. And then typically it stops there. So even when our blood is deoxygenated, it's still over 70% oxygenated, which is why that venous blood is still red. So things that affect saturation of the hemoglobin. Temperature changes, again, not super important in the body, but a little bit. Partial pressure of carbon dioxide, hydrogen ions or pH level, and then something called BPG, which I'll address in a second. So if any of these things increase, increasing temperature, increasing carbon dioxide levels, increasing hydrogen ions, which does decrease the pH, or increasing BPG levels, we decrease hemoglobin's affinity for oxygen. So what that means is that less oxygen binds. The opposite is true too. If we decrease any of these factors, we will increase the affinity and we'll be able to get more binding of oxygen. So let's look at that. So here's the curve again. And when we increase those factors, we do something called shifting the curve to the right. So now we can't achieve as high saturation for the same amount of partial pressure. And then if any of these factors go the other way, we shift it to the left. But that's not as important for normal function. Now, the interesting thing is that all of those factors, especially changes in pH and carbon dioxide and BPG, they are likely to increase at the tissues. So at the level of the tissues, we have more carbon dioxide because the tissues are producing it. That's going to lead to more hydrogen ion levels, so a decrease in pH because of the carbon dioxide. DPG is a metabolic product, so the tissues are metabolically active, it produces more of it. And these things weaken the bond between the hemoglobin and the oxygen, which makes it easier for the oxygen to come off of the hemoglobin and move down its pressure gradient into these tissues, which need the oxygen because they're using it. The shifting based on the acid is called the Bohr effect. It's just some guy named Bohr that described this change in the curve based on acid levels, so that's where that comes from. This is showing you specifically the pH. So acid conditions like occur in the tissues mean that oxygen moves into the tissue. So this is favored by the conditions with internal respiration. Okay. And this doesn't usually happen, so don't worry about that. BPG is, like I said, a metabolic product, so it means that the tissues are using oxygen. And so again, it's nice that it favors the movement of oxygen off of the hemoglobin into the tissues. If you're curious about it, that's the Wikipedia link, but you don't need to know anything else about it other than the fact that it's called BPG and it shifts the curve to the right. This is showing you temperatures. When it's hotter, we weaken the bond and oxygen moves off. And when it's cooler, we lose it. But we're not usually having very big temperature changes. So this isn't super important, except in extreme cases of like fever, hypothermia, stuff like that. This takes us to how we carry carbon dioxide in the blood, which is a little bit different than oxygen, of course. So there's 3 ways that we carry it. Primarily, we're carrying it converted to bicarbonate. So I told you that carbon dioxide will combine with water to form an acid, carbonic acid. But carbonic acid actually likes to break down into hydrogen ions and bicarbonate. So most carbon dioxide gets converted in the blood all the way to bicarbonate, which also means we're going to have hydrogen ions. A small percentage are able to attach to the protein portion of hemoglobin, which makes carbaminohemoglobin, and then some of it is dissolved in the plasma. You can see it's a larger amount than the oxygen, and that's because of the higher solubility of carbon dioxide versus oxygen. But still, bicarbonate is by far the largest component of carbon dioxide transport. Interestingly, regardless of the carrying form, most of it is still transported in red blood cells. So red blood cells might be all about oxygen, but they actually transport most of our carbon dioxide as well. And that's in part because hemoglobin is the major protein for binding to carbon dioxide. Technically, other proteins can bind it too, but it's really mostly this carbaminohemoglobin. And also because the enzyme that favors the reaction of carbon dioxide into carbonic acid into bicarbonate is present in high quantities in the blood cells. So the red blood cells have carbonic anhydrase. It's the enzyme that converts it. This is a fully reversible reaction too, of course. So when we need to get rid of carbon dioxide in the lungs, it goes back to carbon dioxide and we breathe it out. Because we're also making hydrogen ions here, we're triggering that Bohr effect, which releases oxygen from the hemoglobin. And then once we convert it into bicarbonate, it pops into the plasma. It exits the red blood cells and pops into the plasma. Because it's negatively charged, the red blood cells tend to pick up chloride ions to keep their charges balanced, which we call a chloride shift. So there's kind of an opposite effect called the Haldane effect. So remember that oxyhemoglobin tends to favor oxygen binding to hemoglobin? Well, deoxyhemoglobin tends to bind carbon dioxide better than oxyhemoglobin does. So when we have a lot of try that again. Okay, so when we get to the internal respiration at the tissues, right, we want the oxygen to leave the hemoglobin and go into the tissues. We also want carbon dioxide to come from the tissues and start loading up, you know, onto these hemoglobins. As we are converting carbon dioxide into bicarbonate, we're also making those hydrogen ions. The hydrogen ions cause the Bohr effect, where we're unloading the oxygen so that more carbon dioxides can bind, because carbon dioxide binds to deoxyhemoglobin better than oxyhemoglobin. And the deoxyhemoglobin also binds hydrogen ions. So it helps neutralize the acid that's being created as we're getting more and more carbon dioxide into the blood. So because we have oxygen moving into the tissues and we have less oxyhemoglobin, it really favors carrying the carbon dioxide at the same time. So this is called the Haldane effect. So the cool thing about this is that the Haldane effect reverses when we get back to external respiration. So here's the Bohr effect. We've got acids and bicarbonate being produced. This is helping the oxygen come off and move into the tissues, right? So then we have the deoxyhemoglobin. It goes to the lungs, and we're picking up a bunch of oxygen. So we want this oxygen to bind to the hemoglobin. As we bind it to the hemoglobin, we're losing the carbon dioxide off, and we're also taking the acid off and converting these guys back into carbon dioxide, which we're moving off the lungs. So the Haldane effect and the Bohr effect are really nice examples of how this entire system is set up so that the things that are happening anyways favor them happening. If that makes sense. I know that kind of sounds like a circular thing, but it works really, really well. So this is a nice summary of carbon dioxide transport. Carbon dioxide moving in, getting converted into bicarbonate, which then is traveling in the plasma. We also have just dissolved carbon dioxide in the plasma, and then some of it stays in here bound to hemoglobin proteins. One last word on this. So carbon dioxide, because of this association with hydrogen ions, acts as something called a buffer. So a buffer is a molecule that helps maintain constant pH levels in spite of changing levels of hydrogens or hydroxides. So when we have hydrogen levels going up, we get a decrease in pH, so more acidic. And when we have hydroxide levels going up, we get an increase in pH, so it's more basic. So this whole set of reactions, the water plus carbon dioxide, which reversibly can convert into the carbonic acid, which can reversibly convert into hydrogen ions and bicarbonates. It allows the blood to not have its pH changed even if we're having changing levels of hydrogen ions. And that's because you know, if we get higher levels of carbon dioxide, reactions move down their gradients too, their concentration gradients. So this drives the equation this way, and we create more hydrogen ions. But if hydrogen ions levels go up, then we'll see more of these guys combining together, driving it back into carbon dioxide. And so this is where the acid-base levels, where the pH monitoring driving changes in respiration makes a lot more sense, because if our acid levels are going up, we want to get rid of more carbon dioxide at the level of the lungs, and we can just keep converting the bicarbon, the hydrogen ions into carbon dioxide, and then breathe it out and get rid of it. And we'll tackle that more eventually when we talk about fluid balance, but I just wanted you guys to kind of be aware of that, and then look back at how pH and carbon dioxide are driving our breathing, okay? Because that kind of all ties in together. So here's the summary of how that works. So respiratory changes can alter blood pH by altering carbonic acid levels. So when we're not breathing as much, so slow and shallow breathing, we increase the carbon dioxide levels, which drives that whole equation to the right. So drives it this way and makes more hydrogen ions, and then we're decreasing the blood pH. The other way around too, so if we're doing really rapid deep breathing, we're losing a lot of carbon dioxide, more than we should, and that drives the equation back to the left. So as this leaves, everything moves this way to try to keep all of this in equilibrium. And so then we're losing hydrogen ions, and that's going to increase the blood pH. Okay, so we can be acidic over here or more basic over here. There's a couple of resources in carbon dioxide transport if what I said didn't make sense. And yeah, that's the respiratory system. We'll do immune system in a nutshell on Wednesday. Does anybody have any questions? All right.